Good morning. It's February 19th and this is your daily brief in science. Here's everything you need to know. A new method called Ribo-STAMP is opening a window into how proteins are made in single brain cells. Published in Nature on February 18, 2026, the study maps translation across nearly 20,000 cells in the mouse hippocampus, revealing cell type and isoform-specific patterns that do not always match mRNA levels. By tagging ribosomes and sequencing RNA edits with mas-ISO-seq, researchers linked translation directly to full-length isoforms at single-cell resolution, overcoming limits of previous short-read or bulk tissue approaches. Presynaptic versus postsynaptic translational profiles differ, with presynaptic genes more engaged at baseline and after BDNF stimulation, and certain long CDS and 5'UTR features tied to translational control in these compartments. In CA1 subclusters, high translation groups align with bursty firing and mitochondrial gene enrichment, suggesting distinct translational programs related to neuronal activity and energy metabolism. Isoform-level analysis shows edit-C correlations are often negative or absent when shifting between protein coding and intron-retaining isoforms, indicating a separate regulatory layer for translation. Neuron-specific ELAV-like proteins bind extended 3' UTRS of highly edited isoforms and promote translation by interfacing with the translation machinery, as demonstrated in CADM3 isoforms with differential translation. The findings point to potential relevance for autism, Fragile X syndrome, and tuberous sclerosis complex, where altered protein synthesis is linked to synaptic dysfunction. Isoforms with longer regulatory regions tend to drive higher translation, linking transcript variation to protein output and disease relevance. Longer 3' UTRS come with more miRNA binding sites and lower GC content, suggesting a cis-regulatory architecture that supports neuronal translation. Overall, longer regulatory regions accompany increases in translational efficiency, indicating isoform choice modulates protein production in neurons. The study addresses how translation relates to brain disease, why memory neurons differ in translation, and how isoform-specific translation informs disease understanding. A global study reports a dramatic rise in days with weather conducive to wildfires, and the trend is consistent across continents. The analysis published in Science Advances shows the number of days with hot, dry, and windy conditions has nearly tripled over about 45 years, from roughly 22 days a year in the late 1970s to early 1990s to more than 60 days in 2023 to 2024. In the United States, such synchronous fire weather days rose from about 7.7 per year in the late 1970s to 38 days annually in the last decade, while southern South America saw an even sharper jump from 5.5 to 70.6 days per year, with 118 days in 2023. Researchers attribute more than half of the global rise to human-caused climate change driven by fossil fuel burning, based on computer simulations comparing the current period with a fossil fuel-free scenario. The team's simulations indicate that over 60% of the increased fire-conducive days would not exist without increased greenhouse gas emissions. The study concludes that human-caused climate change is the primary driver behind the global rise in dangerous fire weather days, though it notes that weather is a key factor but not the sole determinant of fire impacts. Other elements like available fuel, oxygen, and ignition sources also shape actual fire outbreaks. Synchronous fire weather can overwhelm firefighting resources and complicate cross-border aid as neighboring areas face simultaneous risks. Lead author Kong Yin and co-author John Abatsoglu stress the importance of understanding that weather is a key factor, but not the sole determinant of fire impacts. A 228-meter-long sediment core pulled from beneath Crary ice rise in West Antarctica marks the longest such recovery from under an ice sheet and carries a 23-million-year climate record. The multinational team, working with Antarctic New Zealand and the U.S. National Science Foundation, lived in tents and conducted the drilling with 29 scientists and support personnel, highlighting the scale of cross-continental cooperation required for this work. Samples are en route to New Zealand for analysis by researchers across 10 countries, aiming to determine past temperatures and melting conditions relevant to future sea level rise. Preliminary dating uses marine microfossils, with plans to apply multiple dating techniques to refine the timeline over a 23 million year span. Certain sediment layers contain shells and marine remnants, indicating periods when the region was open ocean and the ice sheet was temporarily absent. The core sits near potential breakup pathways toward the Ross Ice Shelf, about 700 kilometers from the nearest Antarctic station, underscoring logistical challenges. The operation provides a long environmental record, including warmer than present periods, to inform how the West Antarctic ice sheet may respond to warming temperatures. Findings ground truth past open ocean conditions in the region and help constrain models of ice sheet stability and potential sea level contributions in a warming world. The project aims to inform debates on climate policy, sea level rise, and the stability of Antarctic ice under future warming scenarios. Preliminary dating suggests the record spans the last 23 million years, capturing warmer global climates and guiding predictions about ice sheet retreat as temperatures rise. A diagnostic aid called DeepRare analyzes clinical data, genetic information, and literature to generate ranked diagnostic hypotheses for rare diseases, with reasoning tied to verifiable medical evidence. It connects symptom patterns to underlying causes through specialized tools and knowledge sources, aiming to improve accuracy in identifying rare diseases. The article situates DeepRare within a scientific context that references rare disease genetic studies, clinician workflows, and AI-assisted diagnostics. Authored by Timo Lassman and noting affiliations with the Kids Research Institute Australia and the UWA Centre for Child Health Research, it also acknowledges Anthropic's Claude Opus 4.5 used to prepare the News and Views feature. Robustness is demonstrated through cross-center evaluations, specialty-wide analyses, performance on tail-end disease cases with recall at 1 greater than 0.8 for about a third of rare tail cases, and ablations. DeepRare is deployed as a web-based diagnostic co-pilot for clinicians. The piece indicates access to nature journals and provides DOI references, signaling a scholarly feature rather than general audience content. Evaluation spans multiple datasets totaling thousands of cases across in-house and public sources, covering hundreds of diseases and several specialties, with some cohorts including whole exome sequencing. The work sits within the broader literature, citing prior Nature papers and reviews to frame the significance of AI-assisted diagnosis. The piece highlights the diagnostic odyssey for roughly 300 million people with rare diseases, often spanning 5 years or more with misdiagnoses and unnecessary treatments. DeepRare relies on a 3-tier MCP-inspired architecture, a central LLM host with memory, specialized agent servers for phenotype and genotype analysis and knowledge retrieval, and outer-tier web-scale medical resources for evidence. 10 physicians reviewed 180 cases, finding a 95.4% average reference accuracy for DeepRare's reasoning and evidence chains, indicating high reliability of the outputs. Experts evaluating DeepRare's reasoning and references reported correct information 95% of the time, supporting the tool's clinical relevance. A new study led by Professor Juan R. Parilla shows HIV-1 integrase forms an RNA-binding filament inside the capsid lumen, anchoring viral RNA to the core during maturation. This reveals a structural role for integrase in early viral assembly, challenging the idea that its function is limited to later stages. The filamentous IN structures link RNA to contacts with the capsid protein, suggesting IN-CA interfaces participate in core assembly and RNA retention. Disrupting in LegF/P75 binding sites on the IN-dimer interface overlaps with alanine targets, and such disruption reduces infection while increasing eccentric particles. That points to a therapeutic angle targeting IN-CA interactions beyond traditional replication inhibitors. The work reflects a broad, multinational collaboration spanning the Francis Crick Institute, Dana-Farber, Oxford, Birkbeck, Harwell, Imperial College London, and Harvard, supported by NSF, NIH, and the DOE. The study builds on multidisciplinary efforts funded by major public agencies, underscoring a collaborative research ecosystem. Previous research notes, alanine inhibitors can disrupt integrase oligomerization and destabilize integrase capsid interactions, suggesting targets beyond standard approaches. Allosteric integrase inhibitors disrupt oligomerization and its RNA and capsid interactions, highlighting potential therapies outside FDA-approved drugs. Functional validation shows mutations that disrupt N-CA contacts or NTD stacking severely impair infectivity and alter particle morphology, with some compensatory mutations restoring function via IP6 interactions. The ion octamer consists of two CTD-containing tetramers bound to RNA, using conserved positively charged residues to contact the RNA backbone, while NTD interactions connect the octamer repeats. Parilla stresses the ongoing need for new HIV therapeutics and frames this discovery as a stepping stone toward novel inhibitors suitable for clinical use. Cryo-EM imaging required rapid freezing, precise control of the environment, and extensive computation to model capsid integrase interactions from millions of two-dimensional images. This has been your Daily Brief in Science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.